I don't feel like
Más o menos. Sí, cada slide. Y no se olvide que se controla, pero es adelante para pasar la slide. Sí, vamos a tener partido.
You know that I live with people but what is it?
Sí, es por si acaso, ¿no? Por si... Okay, so let's start the new session. It's my pleasure to introduce you Marc, Garcia, and Ferrán Feixas. They are coming from the University of Girona. Now, this is Girona for sure. And yeah, they will talk about free energy methods related to molecular dynamics. And there are two talks of 45 minutes with a small break of 3-4 minutes in the middle. Okay. And then we have, as you know, the coffee break and the computer exercises. So enjoy. Okay, so thank you, Juanjo. And well, it's a pleasure to be here. And well, my name is Ferrán, and I will start, and then Marc will follow. So the idea is to do a short break between the two talks, because otherwise it will be probably too much. But I mean, so the question is, what do you expect about this lesson? No? I mean, what are free energy calculations? Probably it's everywhere. No, you can find or you need to calculate the free energy. Probably in all the lessons, no, the free energy is something that is going to be mentioned. Okay, so here basically what we are going to do is we are going to focus on the application of these free energy methods in 3 different contexts. Okay, so we will start with some really basic concepts. Okay, and more importantly, we are going to focus on molecular dynamics simulations and which are the limits of empty simulations in order to calculate the free energy. Okay, so this is going to be focused on molecular dynamics and the applications of molecular dynamics to calculate the free energy. Then we are going to focus on how to calculate the free energy accurately of a ligand that binds to a protein. That is a typical example of a process that involves a free energy change. Then we are going to move to a more complex process because, well, you probably know that proteins are in constant motion. So, well, there are several conformations of the protein that are possible. So there is what is called the free energy landscape of proteins. Okay, so we are going to explain you some concepts about what is the free energy landscape of proteins and how it's possible to characterize this free energy landscape. In the exercise in the afternoon, in the evening, we are going to do one exercise where we are going to characterize the free energy landscape of a complex biological process. It's an allosteric transition. And we are also going to apply a accurate free energy method to characterize the binding of a ligand or several ligands to a protein. But the idea is that at the end of the talk, we are also going to discuss a little bit some applications of this free energy landscape concept to the field of enzymatic catalysis and protein design. Okay, so this is a little bit the idea. So basic concepts, protein ligand, free energy landscape, and then some applications. So if you have questions, you can ask during the talk, you can ask afterwards, or you cannot ask, no, but so feel free to interrupt when we are talking. Okay, so if we now we are going to focus more on biology or biochemistry or whatever we want to call it. So if we think about biomolecules, so biomolecules are constantly interacting, associating, dissociating. In some cases, when the biomolecules interact, there is a reaction. Okay, so there are several things that happen. And what is important is that free energy is really important to determine the outcome of these processes. So for instance, if we think in molecular biology, so for instance, there is a free energy change when there are molecules that cross through membranes in the process of ligand binding, when a protein experiences a conformational change, or when proteins fold. Okay, so there are a lot of different processes that involve changes in free energy. So it's really interesting to have methods or ways to characterize these free energy changes. So this is not only important, let's say, for biology itself, but if we want to understand protein function or molecular basis of disease, free energy calculations will be important. If we want to apply or design new drugs, also the concept of free energy will be important. And as you are going to see at the end of this talk, this will also be important when we are trying to design a protein or an enzyme. Okay, so the idea is to show you how we can use these methods in order to do these kind of things. But, so ideally, I mean, it's really interesting to calculate the free energy change of a process. But if you now think in biology or in cells, there are several things that are going on. So experimentally, it's really difficult to calculate or to estimate the free energy of a process, especially when we are trying to calculate something that occurs in vivo. Why? Because the cell environment is super complex, and there are a lot of events that are the ones that you can see here that occur at different time scales. Okay, so there are a lot of processes occurring, and if we want to focus on a particular process, this is like super complex from the experimental point of view. Okay, so because of this complexity, this is really difficult to characterize. So the idea is we need methods that allow us to estimate these free energy changes. But the environment of the cell or the environment of biological process is not the only thing that is complex, because if we want to estimate a free energy change that involves a protein or a molecule, I mean, you saw yesterday in the molecular dynamics session that molecules are constantly moving, right? And there are some motions, like for instance, a bond vibration takes place really fast. But if we now focus on proteins, well, there are several things that can move. For instance, the loop of a disordered region of a protein can move. And this can be really fast in the nanosecond time scale, but also can be really slow in the millisecond time scale. Protein folding, it's quite slow in biological time scales. This afternoon, we are going to also discuss something about allostery. Allostery is also a slow process. Okay, so when you think in a protein, you have to think that this protein is constantly moving, and also that there are conformations that are more stable than others. Okay, so if we want to estimate the free energy of a process, for instance, of a ligand binding to a protein, we need to consider that the ligand and the protein are moving. Okay, so these are important things to consider. But if these conformational changes or motions are slow, this is also going to be something that we should take into account when we want to estimate these free energy changes using computational methods. So this is the idea, no? So what you see here, this is what we are going to discuss next. This is a molecular dynamics simulation that simulates the binding of a ligand into the active site of a protein. Okay, this is a super model system of ligand binding. This protein is trypsin, and the ligand is benzamidine. So if you want to look for people that study protein ligand binding, this is always the model system. Okay, but the idea is, so now let's focus what happened in reality. So we have a receptor, we have a ligand, and then this ligand interacts with the receptor, and they form the complex. Okay, typically, if a ligand is a good binder, this process is going to be faster, no? The K-on is going to be faster than the dissociation. And here is where we can define the equilibrium dissociation constant, or the equilibrium binding constant if we do the opposite. Okay, so this is a process that is in equilibrium, so we can have like multiple binding and unbindings, but if we want to design, let's say, a good drug, it's better to have a slow K-off, because we want that the drug remains as much as possible forming the complex with the protein. Okay, so the idea is, no, in thermodynamic terms, if we have this situation here, no, we have receptor, the ligand, and then they form a complex, and we know the dissociation or association constant. So from this, we can calculate the free energy of this process. If this process is net, so the free energy of binding, no, that describe this situation here with respect to this situation here. If this process is negative, this means that this is favorable. Okay. Of course, no, if we talk about free energies, this free energy can be decomposed, and typically, no, we decompose it on enthalpy, entropy. Okay, so well, we can also try to measure these kind of things, but we can further decompose these terms, because if we now think about a ligand binding to a protein or to a receptor, you agree, no, that here we have water molecules. Are the water molecules contributing to the free energy of binding? Probably yes, right? So we have hydration, we have interactions, we also have conformations. This is also translating and rotating. So all of these are aspects that if we want to calculate accurate free energies, we need to consider. And spoiler, I mean, the problem is that it's impossible to consider all these kind of things. Okay, so we are going to do some approximations. Let's focus about entropy, that it's something that we are not going to discuss like in depth here, and about water. No, because when we have like the complex here, no, so here we have like the protein with the ligand bound. And here we have the protein with the ligand unbound. Okay, so this is the process that we are studying. So now let's focus on the active site. So typically, or the binding site. So here sometimes we have water molecules. These water molecules need to be displaced by the ligand. Do you think that these waters want to live? If you think in terms of enthalpy, so enthalpy is what? Interactions, right? Okay, so in terms of enthalpy, they don't want to live, but in terms of entropy, yes, no? So there is always this, no? I don't want to say compensation because it's not so this is like a super discussion, but no, there is always this trade-off between enthalpy and entropy. So when you study a process, you don't have to focus only on understanding which are the interactions that the ligand is making with the receptor and estimating the free energy from there, because there are much more things going on. And there is one important thing that is often, or I don't want to say neglected, but not considered a lot, that is the entropy of the protein. Okay, because if you now think a ligand that is interacting with a protein, in terms of conformation or dynamics, what do you think that is happening to the protein? So the ligand will make more rigid the protein or more flexible. So intuitively, but this is not always the case. Okay, because entropy also plays a significant role. And there are a lot of molecules that bind to proteins that make the protein more flexible. And this is, for example, a case, sometimes this happens in protein allosteric. And this enhanced flexibility is what triggers function. Okay, so this is something to keep in mind, that the conformational entropy of the biomolecule is also really important, especially when we have like disordered proteins that we want to target with a ligand. Okay. Well, I mean, the idea is that you have the presentation, right? I mean, I don't know if you have it yet, but there are like several references here that we are not going to discuss, that if you are interested in some topics, you can come here and look at the reference. Okay, so now, well, it's really interesting to estimate the free energy of binding, but now let's go to, okay, so how we can quantify this? And we want to quantify this with a molecular dynamics simulation. So from the point of view of statistical thermodynamics, we can relate the free energy with the partition function. Okay, so this is expression here is similar to the one that we saw before with the equilibrium binding constant, but here is the partition function. Okay, so this is, I mean, we are not going to talk a lot about statistical mechanics, but this is the basis of the things that we are going to do. But, okay, so here is the expression of the partition function where we have also this Boltzmann probability density functions. But what I want you to keep in mind is that what we have here is that is related with the partition function is the probability of a particular configuration. Okay, so when we have our system, though, depending on the interaction between the molecules in our system, we are going to have like different configurations. So here are like different XYZs. Okay, so for each configuration, based on the Boltzmann distribution, we can calculate which is the probability of this configuration in this ensemble that we have. Okay, so there will be configurations that will be more stable and configurations that will be less stable. And what is more interesting is that we can calculate which is the probability of a particular group of conformations. Okay, and now think in terms of ligand binding. You agree that the protein and the ligand can be bound or unbound? So if we calculate the probability of B unbound and the probability of B bound, do you agree that we will have information about the free energy of the process? No, so this is something that you have here, right? So if we define the free energy difference between two states and imagine that this is the bound state and this is the unbound, the only information that we need from the statistical thermodynamics point of view is the ratio between the probabilities. No, this is simple. So we need to basically count the number of times that we are here and the number of times that we are here. So now let's think in molecular dynamic simulations. Do you agree that in molecular dynamic simulations, we can simulate the binding of a molecule and the unbinding of a molecule? Theoretically? Now don't think about sampling, that is going to be the next thing that we are going to discuss. But from the theoretical point of view, I mean, yesterday you prepared an MD simulation probably, more or less. So when you prepare your system, you prepare your biomolecule, your protein, and you can put the ligand, let's say, in the binding site, and you can simulate how the ligand is interacting with the protein, but you can also put the ligand outside and simulate the diffusion of the ligand. The ligand will explore a lot of different things, and eventually at some point should find the binding site. So this will be binding. If we keep going with this MD simulation, after a while, that it's going to probably going to take years, no? We are going to see the unbinding. But theoretically, this is correct. So in principle, we can simulate binding and unbinding. So from the point of view of an MD simulation, imagine that we have an infinite MD simulation. In this infinite MD simulation, we can capture a lot of binding and a lot of unbinding events. So this means that from these MD simulations, we can count which is the number of times that we are bound and which is the number of times that we are unbound. And from this, we can get the ratio of probabilities, and from this ratio of probabilities, we can get the free energy of binding. Okay, so that's the idea. Okay, so this is what I was explaining to you here. Basically, from the ratio of the probability of the bound state with respect to the separate state, using this equation here, we can calculate the free energy of binding. Since we are at constant volume, the Helmholtz and the Gibbs can be approximated to be similar. Okay. The only thing that we should consider is that when we are doing MD simulations, we are defining a solvent box. Okay. So in order to correct the free energy, we need to consider this correction for the concentration that we have in the simulation box. But basically, using the probabilities, volume of the box, the Avogadro number, and the standard concentration, we have enough things to calculate the free energy of binding. So this is like theoretically correct, and this is what we can do. So from the practical point of view, and this is something that we are going to do this evening in the session. So imagine that we have this protein that you see here, that is a protein kinase, with all these ligands here. Okay, so here is the same scaffold with different functional groups. And we run this, we place the ligand outside, and we run these simulations in order to observe the binding and unbinding events. And we monitor the distance between the ligand and the binding site along the MD simulation. Okay, so we are going to have like a lot of numbers here that are going to represent the numbers. From these distances, do you agree that we can do a histogram? So the free energy, so if we have the histogram and we apply this equation here, no, from the histogram we can take the probabilities, and from the probabilities we can calculate the free energy. This is the idea, no? So always that you have, I don't know, some kind of data, distances, angles, whatever, that you can do a histogram or a kernel density plot or whatever you want to call it. Then from the probabilities that you have here, you can calculate the free energy. Theoretically, this is correct. But if you now think about the lecture that you did yesterday, is this possible? So what's the problem here? Sampling, no? So it's possible to have a long simulation in order, so long enough simulation in order to obtain accurate histograms, accurate probabilities, and accurate free energies. So this is quite impractical. So why? Because typically the time scale of ligand binding is from microseconds to seconds. And the time scales of ligand unbinding is from microseconds to hours of real time. Eh? So now imagine simulation time. So yesterday when you were doing MD simulations, I mean, you more or less calculated the amount of time that you need to, I don't know, to simulate 100 nanoseconds. No, so probably this with the GPU now can be done in 1 day. No, that is the numbers that you have here, but you can probably not see. Okay, so these are like, so now a lot of GPUs are done, a lot of MD simulations are done with GPUs. Okay, and now with the new GPUs, more or less for a small protein, you can more or less obtain like few hundreds of nanoseconds per day. So now imagine that we want to simulate the ligand binding or unbinding. No? So if we want to arrive to a second time scale, it's almost impossible with conventional MD simulations. Okay, so here we have a problem. So the problem is the ergodic hypothesis that you probably mentioned yesterday. And the idea is that it's unfeasible to sample all the possible events that are going to occur in a single MD simulation. Okay, so theoretically, we can obtain good probabilities from MD simulations, but practically we are limited by the computational cost of these simulations. Okay, so, I mean, this means that, well, we cannot use like conventional MD simulations to estimate good free energies, but, well, there are other options. Okay, because, well, clearly the problem is sampling. Okay, so now we can discuss other problems, but the first problem is sampling. Okay, so the thing is, if we use conventional molecular dynamics simulations, realistically, we are limited to microsecond timescales. But this is something that we also discussed yesterday. There are a group of methods that are called enhanced sampling methods that basically we bias our simulation in a way that we can reach a slower timescales, like milliseconds, for example. And this is something that we are not going to discuss, but now if we combine machine learning with enhanced sampling, so there are ways to arrive to slower timescales, okay? But we are not going to discuss this. Another way to basically overcome the sampling problem, that this is something that we are going to discuss tomorrow, is using also coarse grain MD simulations, no? That are simplified representations of your system. Okay, so there are several options here, but the idea is that in principle, with a conventional MD simulation, we cannot simulate the process of ligand binding and unbinding enough times in order to obtain good probabilities and good free energies. Okay, so this is like the take-home message of this book. But well, what I said is not really true, in the sense that there are computers that are specialized, really specialized to do MD simulations. So I don't know if you never heard about this computer here that is called Anton. This has been specifically designed to do MD simulations. Okay, and if you want to know the story of this computer, then you can ask, because it's a long story. But the idea is that in one day, using this Anton supercomputer, they can arrive to 100 microseconds. So this is something that is really significant. Okay, so using this supercomputer, they did this kind of simulations. So they placed fragments of ligands outside the protein, and they monitor the distance between these fragments and the center or the binding site of the protein. And as you can see here, they sampled multiple times binding and unbinding. They did this with ligands that are millimolar. Okay, so they are not really active. Okay, but using this Anton supercomputer, they were able to properly characterize these multiple times and obtain good free energies. So technically this is possible, but the point is impractical, because here they did the same with a molecule that I think that this morning you also discussed, the imatinib, which is an inhibitor of protein kinases. So they also simulated the binding process. So only to capture one binding, they required 10 microseconds. And actually the residence time of imatinib is super slow, so it's hours. No? So in this case, it's impossible to simulate the unbinding using unbiased MD simulations. There are other strategies that we are also not going to discuss a lot. That so some people, the group of Gianni de Fabricis in Barcelona, they had the idea of taking trypsin, and they run 500 simulations of 100 nanoseconds. So a lot of different simulations, really short. And in these 500 simulations, they captured like 300 events, binding events. So from these simulations, they constructed what is called a Markov state model. And from this model, they were able to estimate the binding free energy. They obtained this minus 5.2 value, and the experimental value is 6.2. So not bad. But again, a lot of sampling, a lot of calculations. Okay, so this is the real program. And if you want to learn more about this, there is this paper of Robin Betts and Ron Dror, basically where they discuss this. So how good are these methods in order to capture the spontaneous ligand binding process? Okay. So ideally, no, it will be perfect if we can simulate with an MD simulation without any restraint the binding and unbinding of a ligand, because from this we can obtain probabilities and we can obtain good free energies. Perfect. So in terms of sampling, if we have like infinite computational power and infinite time, this is okay. But now you can think in other things that are not okay with MD simulations. Do you trust the classical force fields? Imagine that you have infinite sampling time, but do you trust the force fields? Maybe the protein force fields are quite good, but I mean, if you have performed some MD simulations with ligands, are the torsions of the ligand properly represented sometimes? The charges? No, so here we have a lot of limitations. Water molecules. There are water molecules that are kinetically trapped in the binding site. No, this is also important. The way that we prepare the system. When you prepare a protein for classical MD simulations, you fix the protonation states, right? And in ligand binding, they say that in 80% of the ligand binding, so in 80% of the ligand binding cases, let's say, there are protonation state changes. But if we use molecular dynamics simulations, we are not considering these changes. There are methods, there are methods that are called constant pH MD simulations that allow for this change. Of course, ideally, I mean, we can use quantum mechanics with molecular mechanics to simulate this, but again, this is too expensive. Okay, so we need shortcuts, no? So we cannot use like complete MD simulations or conventional MD simulations to simulate this. So when we talk about binding free energy calculations, and more specifically alchemical free energy calculations, basically what we are doing is finding a way in order to be able to obtain an accurate free energy with lower sample, so less sampling than the one that we in principle need. Okay, so the idea is to find a shortcut to calculate the free energy. So everything that I am going to tell is based on this article here, the best practices for alchemical free energy calculations, that is published in this journal. If you want to learn more about this, 50 pages with all the information and potential problems of these free energy calculations. So I really recommend you this article. Okay, but before, let's just start. I mean, which methods do you know to estimate the free energy? Do you read that? You can use like molecular dotting. Is this a free energy?
No, is that an MMGBSA?
Exactly, no, MMGBSA, no? So this is also another method, no? Yesterday you discussed metadynamics. No, with metadynamics, can you estimate free energies? Yes, right? So if we start like with really simple methods, like for instance, the ones that use scoring functions, like molecular docking or things like this, of course, we can estimate the binding free energy, but this is really an estimate, okay? Here, this is something that is really interesting because this is quite new, but using quantum mechanics and semi-empirical methods, you can also obtain really good like scorings for the protein ligand binding interactions. And there is this paper here of the Andrese group where they develop a semi-empirical method that provides really good estimates for the calculation of free energies of binding. But here the protein is considered as rigid, right? So this is a limitation, but they obtain like good correlations with experimental free energies. Okay, so the second option is to use like methods like these MMGBSA, MMPBSA, that are called endpoint free energy methods. Okay, so basically in these methods, we simulate our protein with the ligand, and then from this simulation, we extract the information of the protein alone, of the ligand alone, and of the complex. Okay, so basically we run conventional ND simulations, and then we process these ND simulations in order to obtain an estimate of the free energy. These simulations are really simple to set up, are really fast, but are quite inaccurate when entropy is important, if the quality of the force field is not good, and also in terms of conformational sampling. Okay, so this is a good strategy, but probably if you want to really quantify the free energy of a molecule with really accurate methods, this is not the best option. Or if you want to compare one ligand with another ligand. Okay? Then we have another kind of methods that are the ones that are called histogram-based methods. So this is the same idea as I mentioned you before. Okay, basically, we calculate the probabilities from our simulation, but the main difference here is that we perform enhanced sampling simulations. Okay, so instead of using conventional molecular dynamics, we can calculate using enhanced sampling methods the binding affinity using, for instance, umbrella sampling, metadynamics. Okay. What's the problem of these methods? The problem of these methods is that in order to enhance sampling, we have to define a good reaction coordinate that properly describe the event that we want to study. So for instance, if we want to study the binding of a molecule into a protein, we have to define a good coordinate that describes the binding pathway. Okay, so this is the idea. So these methods are really good because we can basically see, visualize the binding pathway because we are defining a reaction coordinate and the ligand is basically following this pathway. So we can visually see the pathway. But the problem is that sometimes it's really difficult to define a good reaction coordinate. Okay, so this can be done, for instance, with umbrella sampling. This can be done with metadynamics. And especially, there is a method that is really good to estimate protein ligand binding affinities, that is called this funnel metadynamics. So here, instead of defining a simple reaction coordinate based on a distance, the reaction coordinate is this funnel that you can see here. So basically, we drive the ligand from the solvent to the protein, and we let the protein to explore the binding site. Okay, so this is a way to accelerate the binding because we are defining this reaction coordinate, but also the unbinding. Okay, so among the metadynamic methods, the funnel metadynamics is really good for protein ligand binding. And I don't know if you can see here the values, but here you have like experimental free energies for different proteins and ligands. And here you have the ones calculated with metadynamics. And more or less, all of them are in the 1 kcal mole difference, which is also quite good. Okay. Then there are methods that are like quite used in the last year. So the people that was working more in metadynamics recently defined this OPS method that is quite similar to metadynamics. And well, this is also quite good for studying protein ligand binding. Okay, so if you are also interested in using enhanced sampling for protein ligand binding, this one OPS works remarkably good. But just look at these plots here. So these are the free energy of binding using a water molecule that is called PIP3P. And these are the ones that are calculated with a water molecule that are called OPC. So if you compare the correlations, what do you see? Significant difference, right? I don't know if some of you have experience in these water molecules. Which one do you think is better, according to the papers? OPC. Okay. But if we calculate the free energies, I mean, this is for the system, right? No, so the correlation is worse than with PIP3P. Okay, so these are always things to consider. And well, there are ways, there are enhanced sampling methods that where we don't need to define a reaction coordinate. And these methods are quite useful. And for instance, a good method in this group is the accelerated molecular dynamics, and in particular, this method that is called Gaussian accelerated MD. And when we do simulations with this method, we basically flatten the potential energy surface in order to move faster between different states. Okay, so this is a really fast way to accelerate a particular biological event without the need to define a reaction coordinate. So this method has also been used by Yinglong Miao and co-workers to study ligand binding and unbinding. No? So here you can see also that in the course of 2 microsecond simulation using accelerated molecular dynamics, it has been possible to describe binding and unbinding more than 10 times. Okay, so this is like good in order to obtain good probabilities and good free energies. So all of these methods are like really good. But we are going to finish like this section with this. So the idea is, I will explain you the basic concepts. And in the evening, we are going to do an exercise where we are going to do an alchemical binding free energy. So maybe in the next 5 minutes, I explain you the basic concepts. And then in the evening, we will discuss like more specific details about how to use these methods. Okay, so the idea is, enhanced sampling is really nice, but it's also computationally really expensive. So now imagine that you are working in a company and you need to design a drug. No? And you have like a good scaffold and you want to introduce functional changes. And you want to know, based on the original scaffold, which is the best ligand. Imagine that you have a library of 100 ligands that are all really similar. And you want to say which are the best 10. Docking is not good for this, no, because you already have a good molecule, okay? So this is, I don't know if this morning you discussed this, but this is called from heat to lead. No, you mentioned this morning, okay. So when you have a good molecule, you want to improve this molecule, okay? And here is when the alchemical binding free energy is really useful. Because, and this is what is important, this method is really good to calculate relative binding free energies. Okay, so not absolute binding free energies. that can also be used to calculate absolute, but then the values are less accurate, or it's more difficult to do a calculation. Okay, but the alchemical binding free energies are really good for relative binding calculations. And the crazy idea here is that the way that we avoid doing a lot of sampling is by defining what is called a non-physical pathway. So imagine that we have this molecule here and this molecule here. And I ask you, which is the difference here, no? So basically here we have this hydroxyl group and that is not here, right? So do you agree that these molecules are quite similar? So, no, if imagine that we want to compare the binding free energy of this molecule with respect to this one, no? Then this is a clear example of relative binding free energy. So what we are going to do with these methods is basically mutate this molecule into this molecule in order to define a thermodynamic cycle that will allow us to calculate the binding free energy. Okay, so this is what we are going to see. These methods are super rigorous and thermodynamically accurate, but also computationally quite expensive, less expensive than in-hand sampling. Okay? And the main point here is the setup. Okay, so it's quite difficult to set up an alchemical binding free energy. And this afternoon, basically, we are going to use a Google Colab that has been basically prepared by the people of Open Force Field, that I would say that it's probably one of the best options to do this without any prior knowledge. Okay? So the point is, well, this is something that I mentioned, but these are the scalability of the different methods. Okay, so the alchemical free energy calculations are here, no? So these are docking that is really fast, and these are the slow methods. Okay, so well, free energy calculations are slow, but are quite good, as you can see here, for do the lead optimization process. Okay, so the idea is not to do free energy perturbation for like discovering hits, no? That is like the first step. The idea is to use these methods in order to optimize the lead. So the idea is, no? Imagine that now we have this molecule here, and we want to calculate the free energy of binding of this molecule into a host. No, what we can do is what I said in the beginning, like simulate binding and unbinding, and from these probabilities, no, we can basically calculate the free energy of binding of this molecule here. And if we want to calculate the relative free energy, we only need to do the difference between this binding free energy and this binding free energy. So using non-alchemical methods, if we want to calculate the relative binding free energy, we need to calculate this free energy and this free energy. What's the secret of alchemical binding free energies? Is that, okay, why not defining a thermodynamic cycle? And this thermodynamic cycle is defined like this. So here we keep having like the binding of the molecule A and the binding of the molecule B. But in this thermodynamic cycle, we have the mutation or the transformation of this molecule in the unbound state from here to here, and also the transformation of the molecule when it's bound to a protein from here to here. Since this is a thermodynamic cycle, if we are able to estimate these two free energies here, we can also get a good estimate of the binding free energy. And the secret is that it will be much faster to calculate these two than these two. No, and that's the basic idea. What can be done with these methods? So basically, this can be used to calculate a lot of free energy difference, not only between different ligands, because we can also use these methods, for instance, to estimate the relative free energy of a conformational change. Imagine that this is not the case, but imagine that this rotation is really slow. So we can also mutate one conformation to another conformation without the need to simulate the complete process. We can also use this to, for instance, calculate the partition coefficients in different solvents. We can also use this to, for instance, calculate which is the free energy of binding to a membrane. because we are only changing the environment. We can also, and this is also something interesting if you work with proteins or protein design, or enzyme design, you can also, instead of mutating the ligand, you can also transform the host. So imagine that here you have a particular amino acid and you want to see which is the difference in binding affinity when here you have a serine or when you have an alanine. So this is also something that is possible to estimate with alchemical free energies. Here we have like the relative free energy of binding, that is the one that we discussed. But also in principle, we can calculate absolute binding free energies, but this is a little bit more complex. Okay, so I think that with this we can finish. And basically, what we are going to do this evening, the first 5 or 10 minutes of the exercise, is going to discuss how these alchemical transformations are performed in practice. And the idea is that we are going to define these lambda parameters that will allow us to transform one molecule to the other by basically switching off the potential energy of one molecule and making appear the potential energy of the other molecule. Okay, this should be done quite accurately, no? Because if you know, do you have in mind the force field equation? Van der Waals terms. There is a repulsive term, no? What happens if we have one atom over another? No, this goes to infinite. No, so there are things to consider when we are transforming a molecule to the other. Okay, so like practical aspects. And what we are going to do this afternoon is basically discussing these kind of things using the Google Colab in order to do these calculations. See? Questions or break? No, so we can do. Oh, yeah.
When you complete the free energy with these end-state methods like MLDSA, so which fragment of the simulation do you use? Because sometimes the ligand is unbind.
Yeah.
So which fragment do you use?
Well, this is a really good question because the point is that imagine that. So the critical point here is the starting point, and this is important for all the methods because if you start with a docking calculation, do you trust the binding mode? No, this is like so a common thing to do is that when you do these MMGBSA calculations or you use these endpoint methods, you discard the first frames because people say that, well, there is an equilibration because you started with an artificial binding mode and, well, this needs to equilibrate. You discard the first, but if you keep going with the MD simulation, maybe the parameters are not good enough in order to keep the ligand bound. So it's making sense to consider these frames? Probably not. No, so this is something that, I mean, using this as, let's say, as a black box, no, it can be a problem because maybe you are estimating the free energies when the ligand is like going out. And imagine that you have like 10 replicas. So probably you are not going to look at all the replicas. No, so it's always really good to combine free energy calculations with distance analysis or visual inspection in order to decide which are the best frames to select for this kind of calculations, because otherwise it can be inaccurate. And also one of the sources of inaccuracies in this MMGBSA calculations is because of this, because the ligand is like flying in the binding site, and maybe this is not what should happen.
So I mean, right? We analyze the reduced displacement, we take the stable part of the simulation, but I think sometimes we are overestimating the value of the energy because we are discarding the structures where there are more motion and so on. Right?
Yeah, you affect I completely agree, and it's really difficult to use this, especially for hydrophobic ligands. This is even more extreme because since you are not considering polarization and this kind of things, the ligand is like moving a lot there, and maybe this is not the case. But yeah, hopefully, I mean, tomorrow we have a talk about machine learning potentials. So eventually, sorry, I will finish with this.
Additional data that you can find.
Additional data, exactly.
Slides that.
So these are the free energy. So a chemical free energy is calculating with a general Lambert force field. That is the typical, no? And these are the ones calculated with a machine learning potential. At least for this particular case, it improves. So, but I mean, I think that there's a lot of things to do here. I mean, to get accurate results also in this particular, using this particular method. But hopefully this is something that this is also going to, the way that we describe the system, this is also something that is going to be improved in the next maybe weeks, no? Because every week there are new papers with new methods that improve the ones that we are commonly using. Okay. Yeah.
Regarding the simulation time in conventional, regarding the simulation time in conventional indeed,
Yeah.
How we can assess if there are big like problems related to the force field or something, because I am now performing conventional LMD. I try to make them as long as possible. And I've been asked, well, but are you sure that this force field is good enough to do that? Because you were mentioning maybe the parameters of the ligands and so on.
This is something
I
yeah.
basically have a look at the energy and see that more or less everything's fine, but I don't know if there is any other way to check that.
Well, there are ways, and one way is calculating the free energy, because from the free energies you can calculate the uncertainties, and then you can also check how these uncertainties change along based on your simulation type. So this is also a good thing to do, like using bootstrapping, sampling, and things like this in order to kind of evaluate these uncertainties. This is something, but this is something that we can talk also 1 hour about this. But for instance, in the beginning, I mentioned David Shaw, that they do these really long simulations. So I remember a talk. So when I was doing the postdoc, people from the Disho group came to give a talk because he did the postdoc in our group, and he explained that when they were doing really long simulations, after milliseconds, the energy was going up and the system was blowing up. Conclusion: the quality of the force field was not good enough. So I don't know which ones of you have done these simulations and, for instance, have seen that alpha helices form in disorder regions. This is also something that is quite common because there are force fields that basically favor the formation of alpha helices. But the question is, these alpha helices should form or not? So there are cases that there are papers of Lynn Kammerlin that they demonstrate that this alpha helix is important. So sometimes it's an artifact, sometimes it's not. So these are things that are, I mean, really difficult to evaluate. But when you use enhanced sampling, is when you start seeing the force fields problems.
And regarding again force field and weather model, because you showed a slide comparing OPC and 3P. From your knowledge, do you know any important difference between these two models?
So in case that you use amber force fields, if you use amber 19SB, use OPC. This is like but with the other force fields, tip 3P, for I mean, it really depends on the problem that you want to study. But so the point is, there are a lot of papers where they benchmark force fields, so look always for this. Because
I was writing molecular dynamics using OPC and FF19SB, comparing the same system but using FF14 to 3P. And I see that my systems are like more rigid when I use OPC.
Exactly.
And I don't know if this is.
So there are
how to explain it, and maybe you have some hypotheses about the flexibility of the protein and so on, and then when you change to these other models.
Yeah, where there's a paper. There's a paper where they evaluate the folding, protein folding using OPC and tip 3P. So probably the answer is there. So yeah, later if you want, we can look for this paper with us. Any more questions? Miguel.
When I look at this kind of graph you are showing here, so in this machine potential, when you look at the extra input you are
Because we have
that we are putting in this machine potential, but we don't have in the flat potential. Are we discussing that non-determined or is non-determined? What is
my feeling, because is that because these are like the early attempts, that torsions are better. That's my feeling, because here they are using still an M for the non-bonded. So my feeling is that a lot of machine learning potentials, I mean, if you use that the one that, this kind of things, what they solve is torsion as a first thing. But of course, there are like additional layers.
And here, the graph are done with the central liquid, they are not the same time of time. Everything stays, so everything is shown with the potential.
Yeah, exactly. But there are like, I don't know, 20 papers that they do this kind of comparisons with different systems. But and of course, there are like multiple machine learning potentials also. So, yeah, so a lot of things to consider. Yeah.
I have a question about the funnel, the process. You show us important that there is the bound state and the unbound state. But the unbound state doesn't really look like the real unbound state because the ligand should be able to be all around the protein. So how does that work?
So it's really difficult to set up these simulations. And this is something that technically I never did a calculation of thermodynamics, so I cannot help. But technically they try to solve it because they don't want to simulate like pure diffusion, no? That in principle this is what is going to happen. But the problem is that if you are doing this direct binding, so slowly the thermodynamics is still quite okay. But if you want to estimate kinetics, then you will have a problem. Because the binding pathway, so the pathway that you're getting is not a super unconstrained pathway. But in principle is what should happen. But as far as I know about this method, thermodynamics is not a problem, but the pathway will be okay, or the kinetics will be okay. So if you try to estimate residence time and things like this for this, they try to solve it. Now I don't remember, but this is a critical thing. Yeah.
Asking another of the methods that you mentioned in Darwin, for example, we said that we didn't need any reaction probably or anything. Just to kind of calculate the person with the energy. So what's the answer? What's the problem with that?
No, imagine that you need to find the right inside. So this can take also forever, even for an also one. So the main point is that this works when you try to use a protein that its the binding site is quite easy to identify. So that you have to charge on the ligand, charge that it also charge amino acids, but otherwise, you are exploring the theories, and you also it's just slower than using a reaction coordinate, but you are not putting any constraint. I would say.
So within these two water models, OPC and PPP, is it a must that we should use OPC during the protein simulation or only during the disordered protein simulation?
Yeah, so that's not a recipe. So yeah, so some people say that that OPC is better, but as you saw, it really depends on the system and the problem, and you can also not compare like 10 force fields and 10 water molecules in each product. So yeah, the point is that if you see strange things, then maybe it's good to double check. What's
the difference between OPC and PPP?
Well, the way that you describe the water, the charges that use extra points to
suppose 3P has 3 points. Yeah, OPC has
no points. Yeah, no points. You simulate the lone pairs and
is it equivalent to 4P or 5P or?
No, so it's a different parametrization scale. So like there's the family, you start with one and you keep modifying, but then the OPC is like a different but again water, like yeah. Sorry, because yeah.
I think I have 2 questions. So, where would you use fundamental methodologics instead of like
volume-based thermodynamics?
So why funnel?
Yeah, why funnel? Is it like just having the state of size?
Yeah, because the basically you are directing the ligand into the binding site. So you are avoiding the.
With volume-based thermodynamics, you know.
You can do.
With volume-based.
Yeah. So yeah, it's the same idea.
Okay.
Yeah. So it's so the idea is the same. It's the funnel. I mean, it's because it's a funnel, but you can also define a volume. Yeah. So you can put whatever the collective variables that you want, but it's the same idea.
Okay.
It's to avoid this oversampling of unbound states.
Yeah, but I'm definitely getting volume-based too.
Sorry?
No, I think I misunderstood.
That
What is to avoid at?
no, like a volume or funnel compared to other metadynamics. So at the end you want to convert these simulations, so.
And in the Gaussian accelerated MD, on what basis do you flatten the energy landscape?
It's magic, no. No, basically you define reference energy. So based on the reference energy, in each point you calculate the potential energy, because you have a force field, you can calculate. So if the way that you flatten is the farther you are from this reference energy, the higher is the boost potential that you apply. And if you are closer to the reference energy, then the boost potential that you apply is small. So the idea is that you basically raise the minimums and you keep the transition states based on this difference between the point and the reference energy. There's a break and second part because maybe then we can play with the exercise in the afternoon to do everything. So
I'll be sure.
So like 2-3 minute break.
For the second part, we need to be a bit.
I will. Yeah, no,
Take it
in the evening we are going to explain some things that are.
Roughly the coordinate of maybe. Yeah. Bonus.
Cada persona a veces hace la
Estás que ver cómo hacen el experimento. Yo creo que una proteína.
Pero no, tienes una muestra cuando tienes una muestra de la muestra natural solo
Thanks for the insight.
I was thinking now when you were asking
But also, I was there actually.
La falta de equilibrio, ¿no? Hay que cuánta cantidad está. Depende si es un factor idéntico, ¿sabes? Seguramente que entra en pero lo que te va a decir es cuánta cantidad es vale, sí, pero eso se puede calcular. Que sea, no, la binding energy de las dos, no, pero eso es buena idea. Sí, 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 lo está dando. Sí, sí. No, i ragazzi. Questa è una parte della pratica. Sì, 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 esatto. Let's start. Okay, so I'm Marc Garcia from Girona, and the idea now is that we can see how we can apply this free energy concept and these free energy calculations in order to understand and analyze protein function, and we will see also some examples on how to apply these concepts to designing new proteins and new protein functions. So first, let me introduce you what we call this free energy landscape. So the idea comes from something that Peter Wallines introduced in the 80s and 90s, that assumes that proteins can proteins can assume a large number of nearly isoenergetic conformations, so what we call these conformational substates, that can be represented in terms of the energy landscape. So different, very similar conformations, but different between them, that at the end can describe can describe protein motions. So how these conformations between of the proteins can be interconverted one between each other. So at the end, this describes how protein moves, right? How we can go from one minima to another minima. So following this concept, we can represent this free energy into these free energy landscapes
that can have very different shapes. And these shapes provide a lot of information. And this is why we want to characterize this free energy, for example, using molecular dynamics simulations, what Ferrán earlier introduced. What I mean with the shape of the free energy landscape. So for example, the depth, the deepness of the potential well, this gives us an idea of the energy, of the free energy, the effective energy that that particular conformation has, considering the unfolded state, for example. But also, when we consider the wideness of the potential well, of the free of this well, we can estimate somehow the structural entropy of the protein. What this means? So intrinsically, how flexible a protein structure is, right? So this free energy landscape can correspond to a normal globular protein, for example. But what happens when we move to a more flexible, more disordered proteins? For example, this case here. So you see that the global minima is not that deep. You see that here the so this is not weakly funneled in the sense that it can explore these multiple substates easily. So going to a more rugged landscapes. So what we find, for example, with a more disordered proteins. So the shape of these free energies are already telling us a lot of information of the conformational dynamics and the structure of these proteins. This concept of the free energy landscape is also very useful to describe protein function. So we know that proteins are essential for, so the free energy landscapes of proteins are essential for molecular evolution, and the change in function can be achieved when we move from one conformational state to another one, for example. So this is what we call the population shift. So it means that we have some pre-existing conformations of the proteins represented in this free energy landscape. And if we populate more one of these less populated originally states, we can move from an inactive state, an inactive conformation, to an active one. Okay, and this change in the conformation of the protein can occur at many different timescales, as we have just seen before, right? So these states much closer in energy are separated by very shallow free energy barriers. So this structural conformational changes takes place at nanosecond timescales, for example. But in this case, we have a much higher free energy barrier. So the conversion from this inactive to an active conformation requires much time. So in this case, millisecond to second timescales. Conformational selection postulates that all proteins can exist in a different pre-existing conformations, and depending on which of these conformations are being sampled, then a ligand, for example, or a substrate chooses one of these states to bind. And after this binding, this population distribution shifts because of the new interactions that are occurring. So at the end, what is happening is that an external perturbation is causing this population shift. I was talking about ligand binding, but also, why not light? Why not a redox electron transfer process? Changes in the pH, or a chemical reaction. A product binds, the conformation of the protein changes, so we redistribute these populations, a reaction takes place, and the protein opens again, so, right? So we have a dynamic conformational ensemble, in the sense that this conformational ensemble of the protein, this free energy landscape, can be modulated by these perturbations. Okay, and this is very important because this can also be applied to understand how the molecular basis for some specific diseases. For example, this is an example. Here in this region, well, this purple colored free energy landscape describes the conformational substates of a particular protein. We see here a native state formed by different conformational substates, and also some folding intermediates and partially folded states. Okay, this can be an example of a prototypical protein. What happened when an external perturbation external perturbation is applied to this protein? For example, in a cell, an increased number of metal ions, a drastic change in the pH. This can induce a complete redistribution of the populations of these substates, and, for example, stabilize amyloid fibrils, amorphous aggregates, so being detrimental for protein function. And in some cases, inducing some diseases. For example, you know, these Parkinson diseases, right? That where amyloids are being formed. So this is induced by a change in the free energy landscape. By external perturbations. But what about, if we can call this in this way, internal perturbation? What if a mutation in the protein structure takes place by randomly in a natural protein, or in a protein that we are modifying by protein engineering, for example? So this is something equivalent. So here is the free energy landscape for the wild type protein, and this is the free energy landscape that we can describe for this particular protein with some phenotypic determinants that corresponds to the most populated conformational state. Okay. In an example of a disease, let's continue with the disease problem. What happens when a new mutation appears? This is what happened with cancer cells, right? Some mutations appear in specific proteins at specific positions, and this induces some cancer diseases. So this is a phenotype induced by a change in the free energy landscape. So now this mutation, which corresponds to a strong hotspot, drastically changes the free energy landscape. So becoming this state 2 now the most populated one. So we change these conformational states and induce this phenotypic response. Also, well, mutations can also induce moderate or weak changes into these free energy landscapes, inducing, for example, these diseases, phenotypes that are not as drastic as these other ones, for example. So at the end, the idea of these changes in the free energy landscape, this population shift induced by external perturbations or by mutations, is the basis for protein evolution and new function design. Okay, and this concept is commonly nowadays being applied in order to design new protein functions. So many, as we just seen, many biological important processes, diseases, enzymatic catalysis, signal transduction, protein folding, allosteric regulation occurs at very long timescales. So it is unfeasible with current methods, Ferrán just introduced this, to sample all the conformational states, so to generate an accurate free energy landscape for all particular system. Of course, with infinite computational time, we could do that. But what if we want to study the effect of a particular mutation? So we should generate, we should generate this mutant protein sequence, and again, redo all this sampling and estimate the free energy landscape and compare and see which is the effect of that particular mutation, if the population shift is that big or not, this is not feasible, right? So there are ways to simplify this, as Ferrán introduced, and we have to consider something here, just following what we have just seen, which is that the sampling problem is more pronounced for rugged and high-dimensional energy landscapes. Of course, when we have when we have a lot of substates very close in energy, for example, these regions here, when we do not have a good sampling, when we are not converging these free energy landscapes, we get really bad descriptions for the free energy landscapes. How to correctly describe, how to appropriately characterize these populations of the different conformational states when we do not have resolution. It is very complicated. And this is why we have all these methods that we briefly introduced. We are not going to review this here again, but there is also an additional problem when we are able to accumulate all this sampling data, which is how to interpret this data, how to generate the free energy landscape from a set of MD trajectories. What I mean, when we run a molecular dynamics simulations, what are we generating? An ensemble of conformations along time, right? How the coordinates of all the atoms of the protein evolve during 100, 1000, 1 microsecond, nanoseconds or 1 microsecond. What to do with all this data? How we can represent all these coordinates for all the atoms in a free energy landscape. Before, Ferrán was talking about reaction coordinates, about collective variables. So this is exactly what we need to generate or to predefine in order to reproduce and study these free energy landscapes. And this is a problem because depending on which collective variable you choose, which coordinate, which degrees of freedom you are defining to create your set of coordinates for your free energy landscape, you will be analyzing one thing or another thing. And this is critical, and this is something that you should be aware of. Okay, what I mean with this? So from MD simulations, we can perform multiple replicas and extract the positions of all the atoms along time. And then we can apply what is called some high-dimensional reduction techniques. For example, principal component analysis, time lag independent component analysis, or applying machine learning tools in order to characterize which are the motions that contribute the most to the conformational dynamics of my particular system, for example, or to the motions that occur at the slowest, that are the slowest ones in my protein. But maybe I'm interested in analyzing a particular motion of one domain respect to another, or when studying a protein aggregation. So then I need to define a variable, a collective variable that describes this movement. And then I can plot this. Later this evening, we will go deeper into this and see how we can transform the MD data into free energy landscapes that can provide us insights on the structure and conformational dynamics of the protein, and use this for understanding how allosteric activations occur, for example. Okay, depending on which problem I want to analyze, I will use one set of coordinates or another one, and this is critical. Okay, so and this is something that you should decide how to analyze your data. Moving on into the protein design and enzymatic catalysis with considering the perspective of the free energy landscapes and free energy calculations. Let's first, very briefly, review how enzyme. Oh, sorry, there are some typos here. How enzymes work. You all know that catalysis, it's about stabilizing, so reducing activation barriers. And what enzyme does is to form first an substrate-bound complex, stabilizing the transition state and generating the product. Okay, and this can be done in many different ways, but once the substrate is bound, strategies that enzymes have to catalyze a reaction is, for example, substrate destabilization. So we make the activation barrier lower, or stabilizing the transition state. So we also make the activation barrier lower. So at the end, we need to estimate how these free energies change. So the free energy of this process change within the active site of an enzyme, right? And this is because we are not going to see all this. This is how enzymatic catalysis works. Well, we just mentioned this, that the enzymes first form the Michaelis complex, the enzyme substrate complex, and then the reaction takes place. Based on this, we can define the Michaelis constant that defines the affinity of the substrate, of my particular substrate, for my active site cavity. And from here, we can derivate the rate, so the velocity of the reaction, so how the rate of the product formation, depending on time. So we can define the velocity of the reaction, define it in this way. Well, this is the Michaelis-Menten equation, which is the basic equation for enzymatic kinetics. I just want to use these concepts here to define affinity of the substrate, so the binding of the ligand, the binding of the substrate, and the catalytic constant of the enzyme, the Kcat, that we can directly relate to the chemical step, so the stabilization of the transition state or destabilization of the reactant complex. So these are two factors that depends on free energies, different types of free energies that we need to take into account when analyzing enzymatic catalysis. And of course, we should consider when trying to design new enzymes. One is substrate binding, the substrate binding event described by the KM. We've just seen before in the first part of the lecture how these binding energies can be estimated. And the second part is the Kcat. So how the chemical step contributes to the rate of an enzymatic reaction. Okay, so here, just not going to go deeper into this, but this is a typical reaction free energy landscape that one can calculate even for small organic molecules, right? So we take reactants, we calculate transition states, intermediate states, and we look for the highest barriers, right, that correspond to what we call the rate determining step, the rate controlling step. These are the bottlenecks of the chemical reactions, and in enzymes, those are the steps that contribute to the Kcat. So this is what we are estimating when we measure in the lab the Kcat. Okay, it relates to that rate limiting step. So for a particular enzymatic reaction, we need to consider which are the highest in energy, so which transition states require have the highest activation barriers. How to calculate this? How to estimate this? For example, for sure, we need quantum mechanics, right? For example, using QM-MM hybrid methods. Not going into this. Tomorrow, the day after, you have a lecture on Friday morning lecture on QM-MM, accurate methods to calculate barriers, to calculate differences in free energies, electronic energies, in different intermediate transition states, different states from a quantum perspective. Why? Because here we are estimating the chemical steps. We are creating or cleaving bonds. We need quantum mechanics, no force fields. Okay? So this is a different a different estimation of these free energies, but also important for estimating enzymatic catalysis. Going deeper into how we can use the concept of free energy landscapes to understand protein evolution, to predict new functions into proteins. The fact that proteins are flexible, that have this plasticity, this intrinsic conformational dynamics described by free energy landscapes, gives them this ability to adopt different states, different conformational states that sometimes allow for functional promiscuity. What it means, functional promiscuity? So it means that if this particular protein can access to a given conformational state, maybe not the most populated one, but accessible, and this conformational state is active for a non-native function, catalyzing a non-native reaction, accepting a non-native ligand. So this is promiscuity. And this is indeed what this methodology called directed evolution proposed by Professor Frances Arnold is based on for designing new protein functions. So we have, for example, this is an example here. We have a particular enzyme that catalyzes a native reaction, but also offers some promiscuous activity towards a new reaction, a secondary reaction, very tiny. But if we introduce changes in the sequence space, if we make mutations into this protein, what we just said, we can change the populations of these free energy landscapes. So maybe this less stable conformational state can become the most stable one. What it means? It means that we are favoring this new function. How? By introducing mutations, by changing the sequence space of this protein. Does this make sense? Okay. So this is how we relate promiscuity and conformational heterogeneity. Because this plasticity, this flexibility of protein scaffolds, we can access to different substates that can be more and more populated. So we can change this, we can induce this population shift by introducing what we call these beneficial mutations, so changes in the protein sequence that stabilize particular substates. We don't know how they look like, or when we apply directed evolution, we don't know how these substates look like, but we introduce these changes in the protein sequence that favor this population shift. And this provides the basis, first, for functional diversity, so why proteins are promiscuous, and a route to evolutionary divergence for new functions, so to create new functions into proteins. And this is what nature has been done for many, many, many years, right? Starting from one scaffold and evolving it, so changing this free energy landscape to access to new functions. This is this can be also described in terms of what it's called the protein fitness landscape. What it means. So in evolution, or in directed evolution in this case, what we are doing is to start from an initial activity towards a particular chemical reaction catalyzed by this enzyme, and by introducing changes in the protein sequence, we can navigate towards we can navigate through this fitness landscape towards desired new functions. So the highest we are going, the more proficient the protein is for that particular function, right? So, and this fitness is the measure of how well a given protein, a given mutant, performs towards a target function. In this case, chemical reactions, for example. Okay, the problem is that we do not know how this fitness landscape looks like. So we don't know which direction should we take. That means which mutations should we introduce, right? That's the holy grail, I would say. Right? And how this is done? Proteins contain 20 natural amino acids. If we have a sequence of 300 amino acids, and we need to test all the different possibilities to see which direction in this fitness landscape are we taking, is this feasible? Of course not. This is not. And this is why directed evolution has this random factor. So you make random mutations in your protein, grow them, you grow these cells, you test your new proteins, and you see, and you screen if the property you are looking for is being improved or not. If it improves your property, you take that mutation. If it doesn't, you discard that mutation. So you navigate this fitness landscape in a random way. But the task for computational chemists is to try to find the amino acid sequence that results in a desired protein structure that is, of course, stable and functional towards your target function in a rational way, somehow. And this is what protein design, computational protein design is about. Try to find or propose strategies that allow us to estimate at the end the free energy of a predicted protein sequence that relates to a protein structure that minimizes this free energy of the system and is able to proficiently catalyze a chemical reaction or to achieve a desired protein property. So we need to define computational strategies to navigate this sequence space intelligently. And this is very complex, as you can imagine. There are different approaches to do this from a computational perspective. We can apply physics-based energy optimization methods. Okay, yeah. So we can apply physics-based energy optimization methods, so MD simulations with force fields, for example, to sample the conformational space to generate free energy landscapes and estimate where the local or global minimas are. So navigate through this free energy landscape. Or a more modern alternative is to use, for example, deep learning-based methods to predict structures that ideally these methods are trained to already predict minimas on this free energy landscape. And very briefly, just introduce you why one of these fundamental, let's say, pioneer works by David Baker were very useful at that moment. The Rosetta-based methodologies, which is a physics-based method that provides this Rosetta energy function that is trained using, you have the slide, so you can check it later, based on this formula here, and is specifically trained to reproduce these native protein structures. And the key of all this is to account for or to predict how the surface amino acids are placed, how the core amino acids are ordered, and also how estimate this boundary region. So residues that are solvent exposed, residues that buried into the protein scaffold, and how this can be treated to correctly predict a native fold protein structure. A lot of successes were achieved by these Rosetta-based approaches, designing new nanomaterials where different proteins are aggregated, also designing binders, designing functional nanomachines where proteins are moving, so you are kind of reproducing protein aggregation and this dynamism. Also, how designing new proteins that change their structure due to external perturbations, what we have seen before. And finally, and then I'll finish with this, is the more challenging protein design, or one of the most challenging protein designing problems, which is designing functional proteins for catalyzing chemical reactions. And this is a and a strategy that was proposed together in the early 20s and 2000 by David Baker in collaboration with Ken Hauck, where they proposed a strategy called Inside Out to design new functional proteins for catalyzing chemical reactions. The idea here is to start from a transition state optimized at quantum mechanics level, so using density functional theory to optimize a transition state that contains a specific amino acid side chains that stabilize that chemical reaction. This is what was called the theoretical enzyme, the thiozyme. So taking this quantum mechanics structure, this transition state, remember, corresponding to the rate limiting step, relates to the K-cut, and create a protein backbone, a protein scaffold around it that can handle these amino acid side chains, and then can be optimized by additional mutations to correctly pack this active site, this transition state, accommodate the substrate in the transition state, and being able to catalyze the chemical reaction. So that was the idea, combining these two approaches, the energetics for the chemical reaction and the energetics, so the free energy of protein stabilization. The problem again is how to choose which of the protein sequences will be able to fold, to be stable, and to host that particular substrate and transition state. The initial designs were based on Diels-Alder reactions, retroaldol reactions, Kemp eliminase reactions. The designs were really poor, and extensive directed evolution were required to make proteins really functional. But the idea was there. And nowadays, this is probably the most successful strategy to computationally design new enzymes. Why? Because now, well, you know what David Baker has been done recently, which is combining this idea with modern AI-based protein designing tools. So using RF diffusion, you can create scaffolds that host a particular backbone conformation. So you can force a protein to be or to have the shape to perfectly match your transition state. You can use also diffusion-based tools like Placer, also developed by David Baker, to account for conformational flexibility of your ligand and your transition state in the active site of that protein. So not just considering a rigid kind of docking structure, but also accounting for this flexibility, for this conformational diversity. And finally, using ligand MPMNN, for example, you can propose protein sequences that stabilize that particular backbone, that minimizes that free energy, making the protein more stable, right? So this is just this is based on a very recent paper that just appeared a few months ago. Maybe, well, we have the information in the slides. We can discuss this later. But just to give you an idea of, from a computational perspective, AI-based tools that aim to reproduce stable conformations, so try to reproduce free energy landscapes, are nowadays being used to design new functional structures. Of course, all this is based on static protein structure predictions, but also, and this is also very recent, the bioRxiv publication paper that has been posted at the end of 2024 by Frank Noé and co-workers from Microsoft, where they propose diffusion-based strategies to predict free energy landscapes, for example, for folding of proteins. Of course, this has a lot of limitations. You have a list here, but just to let you know where the field is going with the idea of reproducing free energies, because at the end, this gives us the information for understanding and designing protein function. Okay, we will end here. We will continue the discussions during the practice lessons after, but we are open to discussions. We're sorry for the time, but
we can continue to the questions after the break, because now we are waiting for the
Sure.
So let's thank Marc for the
Okay, so since we are 20 minutes late, we will come back also 20 minutes later, so we are back here at 5:20.
And we are going to reduce the exercise. We are
Yeah.
That's seven. That's,
Ya lo sé. Si lo sabe mal,
lo contamos con las preguntas del
ya.
Está bien.
Sí. Es más slides o menos. Sí, ya lo van a.
Digamos.